I'm going to present you on the topic today. In the last phase, I delivered a talk on in-silico based drug design technique as part of COMFA based technique like that. So I thought that maybe for this time little bit changes for the second phase, and I modified the title to be Learning Chemistry by Unlearning like that. So you may be wondering what is this title means and what I'm going to say about this particular topic. It is quite an interesting topic. I hope you all will like it, this particular topic, because it is there with everyone in the day-to-day life taking into consideration, then to the chemistry take into the consideration, and as part of the chemistry, the structures, reactions take into the consideration. So it will be, I hope that it will fetches your hearts much better, and I hope that you will find it interesting. Okay, so I'll share my screen. So this the screen is. Am I audible?
Yes, sir. Yes, sir. You are audible, sir.
Okay, okay, fine, fine, fine. Yes. So learning chemistry by unlearning. So
Sir, did you share the screen, sir?
I shared the screen.
But it was not visible, sir. Only you are visible on our screen.
Okay, 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 fine, fine, fine. 1 minute. Yes.
Yes, sir. Yes, sir. Now, okay, sir. Okay, sir.
Yes, fine. So, all of you, this is the topic learning chemistry by unlearning, like that. Okay. So, that means that to learn, first we have to learn whatever we have learned already. So, that needs to be unlearned, and freshly we need to learn. So, in what context I am telling you this? Okay. So, that is what I am going to present before you all. So, what is the public perception of chemistry? As you all know that what is the public perception of the chemistry? Okay, so the perception of the chemistry is that it is called the chemistry causes more harm than the good. This is the general public perception about the chemistry or chemists across the world, if you take into consideration. In what context I'm telling? That means, for example, if you take high profile examples, for example, Bhopal gas tragedy, if you take into consideration. Okay, so Union Carbide company that, okay, in the year 1984, 2nd of December. So there was a gas leak. What was that gas which was leaked in the Bhopal? So it was methyl isocyanate. So very nasty, very harmful, very toxic gas it was. It got leaked from this chemical company, and lot of animals, birds, whole environment got affected. Many people have died. So such is the what the general perception of the chemistry. That means they tend to do more harm with the chemistry than the good, like that kind of an perception like that. Even otherwise, if you think, for example, if you take any chemical companies, if you take around wherever you are there in Hyderabad or any of the places, wherever these chemical companies are there, generally they'll be releasing they'll be igniting the chemicals, and they'll be uh, through the drainages some chemical uh, will be bleached out, and then that chemicals will come to the fields, somewhere it will get into, and they get into affected like that. So if the precautions are not being taken, proper precautions by the companies, so these are all the hazards that can take place. Okay, so but however, everyday items are often not classified as in chemicals. Everyday items often not classed as in chemicals. If you take into consideration, for example, what clothes we wear, what food we take. Okay, so they're all basically and chemicals, but often we don't classify them. Okay, so the benefits of chemistry are not appreciated and can be difficult to put into the context. Okay, so the benefits of this chemistry, because of chemistry research what we do, or because of the chemistry, the production, whatever we do it with that, whatever the outcomes, whatever the products of the chemistry, they are not being that appreciated if we take in consideration. So now if you think that, for example, how many lives have been saved with the introduction of the antibiotics. Suppose if you take penicillin was an wonder drug, okay? So discovered by Alexander Fleming in the year around 1940s, 1930s to 1940s. So now how many lives have been saved because of the penicillin, okay? Not many people will appreciate that. The penicillin is purely a chemical and organic chemical, so it is developed as a drug. So so it was a wonder drug, okay, but not many people will appreciate that, okay. So this is one of the major concern here. Next, if you come back to the history wise, the late 18th century, if you go back, okay, so we are in the 21st century. So the last century, 20th, 19th and 18th, if you go back, okay, so almost 2.5 centuries to 3 centuries if we go back, you can find that at that time the organic means compounds from living organisms it was, okay? So at that time in the late 18th century, it was the concept that compounds from living organisms are considered to be an organic in nature, okay? And compounds from the lifeless matter, it was considered to be an inorganic in nature. Compound from living maybe in plants, herbs, or it may be in animals or any marine source. Any it should be a living source for that. It should be an organic. That was the concept. It was there at that time. And if it is from lifeless matter, that means from the earth, some soil, what we can get it, what are the minerals, what we can get it from the mineral source. So they are considered to be an inorganic in nature. At that time, there was an force, there was a theory called as vital force theory. Okay, so till the late 18th century, the vital force theory was existing. What do you mean by this vital force theory? Okay, vital force theory is nothing but, okay, so it is the theory says that organic compounds must originate from the living source only, okay, from the living organisms only or living source only. So that was the vital force theory was been insisting like that, okay. So till 18th century it was been there like that, but what has happened is that in the year 1828, one famous scientist who demonstrated that ammonium acetate can be converted to urea. Ammonium acetate is an mineral source, it is an inorganic compound, in fact. So this ammonium acetate can be converted to urea. Okay, so just by heating it gets converted to urea. Ammonium acetate will get converted to the urea. So this changed the definition of organic chemistry itself at that point of time, that what they thought at that time, that means organic compounds means they must originate from the living source only, okay. So it was, that was the understanding it was there at that particular time till the 18th century, okay. So then by Wooler demonstrated a pure inorganic substance getting converted to the urea. Everyone changed, modified the understanding of organic chemistry, and they modified the definition of an organic compounds, organic chemistry itself. So like that, what happens is over a period of time, okay, there will be some changes, some modifications, and we'll keep on updating the things. In that sense, what happens is we need to unlearn whatever the things which we have learned it before, and then we have to relearn again freshly the concept so that we can understand better and move forward like that. Okay, so it is a simple thing what I mentioned. Now we'll take it case by case studies like this, what has happened before, um, and and over a period of time how the things have been changed. Okay, like in that context take into the consideration. Okay, so now organic molecules means what do we understand? Organic molecules means carbon based molecules. So, okay, so organic molecules means compounds of carbon. Okay, so they are compounds made up of an carbon chains. We call it as. Okay, so the organic chemistry is about chemistry of the carbon-based molecules. Is all what we study. So some of the properties of these organic molecules are that the stability composed of stable carbon-carbon covalent bonds. Okay, so they are the characteristic features of an organic molecules. Remember, my dear colleagues, actually, so organic molecules today we have around about 20 million organic compounds with us. That constitutes more than 95% of the chemicals that we have all together, organic together. So why there is an explosion of there's so many organic compounds are coming into the okay market as well as to the everywhere it is been synthesized in the laboratory, the academics or in the industry. Wherever we go, we can find okay organic molecules. What is the reason for that? It is reason for that it was because of the covalent bond. Okay, the bond which is formed by sharing of an electrons, it makes it possible to combine one carbon with another carbon, and thereby what happens is these carbon atoms in different permutations and in different combinations they get combined together and build the molecules of our interest line. So because of this catenation property of this carbon compounds, okay, so we can able to synthesize them in a larger numbers like that. When we synthesize large number of these organic molecules, automatically we trying to find out the applications of these organic molecules, and in that way we have found different applications of this organic molecules. So now, if you take a defined molecular structures, okay, some of the properties of organic molecules is that they have defined molecular structures. Okay, so they have a definite molecular structure will be there. Okay, the molecular structure refers to the two-dimensional structure as well as the three-dimensional structures. Okay, so they have a definite molecular structures. So now defined three-dimensional shapes, when they have a structure, so automatically they must possess a definite three-dimensional shapes. Okay, so the three-dimensional shapes of the molecules adopted by these organic molecules, so that will differ from one compound to another compound. Okay, so from one case to another case, it will differ the three-dimensional shapes. Okay, the three-dimensional shape, the three-dimensional structure of the molecule is so crucial, very important that okay. So what you do it in the in silico drug design. Okay, so when you are trying to simulate the molecule for your docking or QSA or for the virtual screening or pharmacophore screening or anything kind of or any kind of tools, then definitely we are going to take the three-dimensional shapes of that organic molecules and we are going to model them. Okay, so next is defined stereochemistry. Because of these carbon compounds and depending upon the chirality of the carbon compounds taken into the consideration. So sometimes the carbon may be of chiral, sometimes it may be carbon-carbon double bond. And because of that, what happens is geometry get fixed. Since the geometry get fixed in the three-dimensional space, so that chemistry we can able to define it like, for example, this is cis, this is trans, this is dextroid, this is levo, this is E, this is Z, this is R, this is S kind of configurations we try to give it like that. So next is biological organic chemicals. So in many of these biologicals are organic chemicals. For example, if you take into consideration, so sugars, okay, all these sugars are basically an organic chemicals. Okay? So most of these sugars are dextro sugars, which are in the biological system. And if you take, for example, proteins, they contain amino acids and they're also biological organic chemicals. And these proteins, amino acids are building blocks for the proteins, and proteins are the building blocks for the cell, and cell is the building block for the protein. So this is how the chain goes. So now if you take these amino acids, which are the building blocks of these proteins, they are of L amino acids and the stereochemistry wise there. Okay, so that's how the nature exists like this. Next, if you take fats and oils, they are also organic in nature. Next, vitamins, if you take into consideration, where it is and water soluble vitamins or fat soluble vitamins. So they basically organic in nature. If DNA or RNA, these are which plays a crucial role in the okay, reproductions DNA and they are basically biological organic chemicals basically taken into consideration. If taken wood, it is also an organic in nature. Natural rubber, if you take into consideration, natural rubber, what it consists of? A polymer of isoprene units. So they are basically a natural constitutes the natural rubber. So many applications of this, and it is basically an biological organic chemical. It is basically. Next, essential oils, for example, if you take into considerations, these essential oils are nothing but an volatile hydrophobic and it should be volatile and they get evaporated uh, where they have been kept. So now, so such essential oils are, even if you take, for example, for essential oils, for example, if you take clove oil, okay, it's an essential oil. So like that, even if you take edible oils are also organic chemicals, what we use it in the kitchen, the edible oils and all. Natural fibers, if you take into considerations, they are also basically an organic chemicals. And the antibiotics, if you take into considerations, all the antibiotics, whatever we have, they are all biological organic chemicals because they are the biological products obtained from fungi. Okay, like that. So basically, these antibiotics are also biologicals in nature, in the sense biological organic chemicals. They are basically. So next, fermentation products, if you take into consideration the fermentation. So now whatever the alcohol, this wine industry, it is going on. So this alcohol has been mainly produced from fermentation. The fermentation products are also an organic in nature. So because alcohol is purely an organic compound. Next, Natural flavors, if we take into considerations, natural fragrances, if we take into considerations, okay, so they're all organic in nature. Natural fragrances, for that matter, okay. So natural flavors, for example, if we take vanilla, strawberry, many of these flavors are available. They are all from the natural sources, but they're all organic chemicals, actually, basically from the biological source. So plants and microbial products, if we take into considerations, okay, plants and microbial products of this, okay, biotransformations and all those things what we study. So they're all basically an organic and biological and organic chemicals altogether. Like biomatter, if we take into consideration, altogether they are organic in nature. So the organic molecules, okay, these chemicals in the manufactured products, if we take into consideration, so bioactive products, if we take into consideration, so what we can find it is the medicines. What are the medicines that we can find it? So in the medical shop, okay, what are the medicines there are, we can find it, they're all chemicals, okay, developed as in drugs. Basically initially it was in chemicals, they have been elevated to the platform of an drugs, like that. After meeting all the standards requirements, they have been elevated to become the drugs, like that. So all these medicines, more than 95% of the drugs which are there in the medical shop, they are organic in nature, okay. So next, if you take even veterinary medicines also, okay, they're also like medicines, they're also organic in nature. If you take herbicides, for that matter, herbicides are also an organic in nature. If you take pesticides to kill the pests, so they are also organic in nature, like. Next, if you take in fungicides, for that matter, they are also organic in nature. If you take in plantations, they are also organic in nature. Fertilizers, if you take in consideration, urea, any fertilizers, what we use it, they are all organic in nature. Such is the importance of these organic chemicals in the day-to-day life, and okay, not many of us will really appreciate the role of chemistry, uh, in this contribution of chemistry as a manufactured products, like that, okay. So now antiseptics, if you take into consideration, for example, like Dettol, like that, so or Savlon, so they're all antiseptics, basically. Next, if you take a disinfectant, your phenyl, what we use it to clean the floors and all those things, they're basically organic in nature. Okay, so all these are the some of these organic molecules in the materials, if you take into considerations. Some fibers, if you take into considerations, fibers and clothing, okay, and then plastics, if we take into considerations, various applications of plastic we can find it today, and coating aids, if we take into consideration. So paper, if we take some packaging industry, it is a very big industry, in fact, for the products to be manufactured and packed and sold it. Medicinal implants, if we take into considerations, for example, your stents and all what has been used, they are basically organic in nature. Some cervical discs, if we take into consideration, those who are suffering from cervical spondylitis or disc collapse, so they'll be using these medical implants. Next, wound dressings and all, if we take into considerations, they are basically an organic in nature, like that. And then even the films, what we use it to develop these, okay, so for visualization and all, they are basically organic in nature. So many applications of these organic chemicals in the manufactured products. So we need to really appreciate the role of these organic chemistry in the form of these products in the market, which are available for us to live the daily life. Okay, like that. So we know normally we were many of the people will not appreciate this. It is all because of the contribution of the chemists. Okay, so like that, for example, into consideration. Now, my dear colleagues, with this brief background about the importance of chemistry and importance of these chemicals as an organic chemicals in the manufactured products, and we are using it all together. Okay, so with this context, I'll come to the some of the case studies where what went in the over a period of time, or okay, how things have been changed over a period of time, like that. Okay, so I hope now this case study will find you some interest, okay, to learn what has happened before. Okay, like some myths, and myths means it is a false belief, and later it has been come to know that this is not the false belief, this is how it is. Then we change it and we correct it ourselves, like that. Okay, so that is why this what we are going to talk is by learning chemistry by unlearning things, like that. So it starts from this slide onwards. So now you can see that, for example, you all know the DNA structure. Okay, it was been given by Watson and Crick, and they have been awarded Nobel Prize in the year 1962. Now, if we look at what has happened in the context of this development of this DNA double helical structure model in the Watson and Crick lab, so what was happening before 1962, around 1955 to 1960, in that period of time, what has happened is that, so what Watson and Crick, they were using the textbook, that textbook was the biochemistry of nucleic acids by J.N. Davidson. It is 1950th edition. It was the latest edition at that point of time, and they were using this textbook, and in that textbook they were being given, you know, that how crucial these nitrogen bases like adenine, thymine, cytosine and guanine, okay, to create this double helical structure, how crucial they are, these nitrogen bases are. If you look at this particular textbook of 1950th edition, they have been given the structures of guanine and thymine like this. Okay. If these are the structures of the guanine and the thymine, remember, my dear friends, no model could have been constructed. It will never comes out to be a double helical structure of a DNA. Okay. But Watson and Crick used these structures of an guanine and the thymine. Okay, so what's wrong with this structure of an guanine and thymine? Okay, so now the thing is that how did the Watson and Crick come to know that there is a mistake in this guanine and thymine structure? Okay, so that is quite an interesting thing. It is that, okay, one theoretical chemist called as Jerry Donoway, he visits his lab, Watson and Crick lab, and identifies the mistake and corrects the this guanine structure and the thymine structure during that particular period of 1955, uh, during that particular point of time. So now if you look at, this is the incorrect structure of an guanine and this is the correct structure of an guanine. What is the correct structure? What is that? What's wrong with this? With this incorrect structure, that means it is an enol form. Okay, actually it was an keto form. You may wonder what makes such small difference like that is just an ketone tautomerism. My dear friends, remember these small things are very important. Unless you, as a chemist, if you don't notice these kind of and small mistakes, so we cannot become a successful chemist. Like that. So a Jerry Dunaway was a theoretical chemist, visits the Watson and Crick lab and identifies these mistakes in these two structures and he corrects it. Okay, so Watson and Crick also acknowledge Jerry Dunaway after getting the Nobel Prize. Okay, like that. So, but Jerry Dunaway knows the actual structure, but he could not uh, come up with the DNA double helix structure, but still it's all depends on how the efforts have been put in place and we are trying to come out with that. Okay, so my dear colleagues, we should have good friends circle that amongst us within the staff members, amongst our staff members as well as with from other colleges also, so that what are the mistakes that we do that can be corrected and okay like that, and there should be a healthy competition amongst ourselves that we can able to uh, do better like that. So that is why uh, it was such an important period of time. At that time, Jerry Donaway corrects the structures of this guanine and thymine and makes Watson and Crick to come out with the double helix structure for the DNA. Like that. So this was the story like that. Okay. So like that we need and another thing is we should ready to unlearn the things and learn the new thing and move forward. So that kind of a mindset or an attitude one should have it. That is very important. Right? Next, another scientist, another case study, if we take in consideration, Aristotle. Aristotle was a famous Greek philosopher in the ancient classical period of Greece. If we take into consideration, he was very famous at that time, a very famous philosopher. And this Aristotle makes a statement at that time that corpora non agunta nisi saluta. It is, okay, it is in the Greek. When it has been translated, it means that substances do not react unless dissolved. Okay, unless these reactants have been dissolved, they will not react like that. What Aristotle says. Now, the question is that how far this is true. Okay. Now the question is that how far it has been true. Okay. So now the thing is that if you take a today's scenario in pharmaceutical industry, today, in the APM manufacturing, okay, so in most of the reactions that we are doing it in pharma industry, even in academics also, is that many of the reactions are carried out as a suspensions. Okay, they are not clear solutions; they are the suspensions. That means reactants are being suspended in the with the solvents like that. So this is what is the today's scenario like that. That means what Aristotle said is that was true? No, it was a myth. Okay, so what was true? It is not necessarily. Okay, for a reaction to happen, the reactants not necessarily should get dissolved. Okay, even it can be like suspensions or okay or emulsions. They can get together and they can form the they can turn out to the reactions can happen, and we have many examples for such cases. Okay. Now, next another example, what I'll take it is, for example, organic reactions needs anhydrous conditions. Okay, organic reactions needs anhydrous conditions. This is one false belief that many of the if we look at many of these our research scholars, what I look at in our lab, when I look at these research scholars, they'll be spending so hard, so much of time on uh, these making these solvents dry and using it like an anhydrous conditions for doing the reactions and all those things. Now the things that is necessary, and another concept is that what most of the students will think it in that is that organic silver solvents are better than the water. This is the general concept. Organic solvents are better than the water. For doing these organic reactions, we tend to use organic solvents in comparison to water. Okay. So now this is the one of the false belief that we have, that it not necessarily that all the reactions needs anhydrous conditions, or not all the times we need an organic solvents to drive these organic reactions. Okay, so why is it so like that? Okay, certain reactions are faster in water itself. Okay, so some of these reactions are really faster in water if we take into consideration. Okay, so one such very good example what we can quote it is that Breslow discovered that Diels-Alder reactions moves faster in water than in the organic reactions. Okay, so this work has been published in the JACS, a prestigious journal, in the year 1980. Okay, by the Breslow. Okay, so till that time, no reactions were being performed. Okay, yeah, like Diels-Alder reactions was being performed using in water. For the first time, Breslow discovers and demonstrates it using in water as a solvent and doing the purely an organic reactions, that is Diels-Alder reaction. Okay, so you know what is the importance of using water. Why I'm stressing on this water is that so since we are whatever the chemistry that we are doing it, if we can make our chemistry green, that means it is on the principles of a green chemistry, we can contribute to this okay, this society or this world. We can make it pollution free as much as it can be possible. Okay, and with our myth, uh, keeping the in the consideration that um, water is not an ideal solvent for organic reactions like that, we should not think in that way. Rather, we have to think it in a way like we can still do many of these organic reactions using in water as in solvents like that. We need to explore them. That is the thing. So now, if we take into consideration this the Diels-Alder reactions, we know that in the Diels-Alder reactions, the diene reacts with the dienophile and it will form a Diels-Alder adduct. It will be formed. In the Breslow's discovery, what Breslow discovered is that when you use the solvent that is isooctane, a purely an organic solvent, the relative rates were there's just one. Okay, when it was an methanol was being used for this reaction to dry, it was 12.5. Okay, slightly increased. That means isooctane is highly non-polar if we take in consideration. From this non-polar to polar if you move with respect to the methanol. Okay, so isooctane has less dipole moment and it is non-polar in nature. If you take the methanol has relatively high dipole moment, and definitely the relative rates have been increased to 12.5. Compared to methanol, we know that water is having higher dipole moment. So then when it is being used water around 1.8 device, it comes the dipole moment and it is quite enormous. And the relative rates have been shifted to 740. Okay, such a drastic increase in the reaction rates, we can observe it like that. So this was because of the as we are using the water as a solvent, which was highly polar, and that was the need of the reaction here for this Diels-Alder reaction, and the reaction rates have been enhanced to such in foods. Take into consideration. And when the water, and along with the water, when lithium chloride is even used, that was even synergistic, and it jumps to an 1800 times. Okay, the relative rates have been increased. Okay, so this will clearly indicates that. Okay, so for this diol reaction, if you are using a more polar solvent, then more the reaction rates, it will be the reason. And also, there is a reason for that why water has enhanced this reaction, this diol reaction, and how this lithium chloride, which has been used in the water, how this has been enhanced this diol reaction. There is a reason, definite reason for it. Okay, what is that definite reason? If you want to know, is that the hydrophobic effect, that means the poor solubility of the reactants in the water. What happens is the reactants in the water, it is poorly soluble because these are dienophile and dienes are organic in nature. They are organic means purely and slightly and non-polar stuffs. So these non-polar stuffs and non-polar substances with the water, what happens is that they won't be getting solubilized. So what happens is they get accumulated. When they get accumulated, water creates some kind of pressures and pushes these molecules together to get aggregated. And because of this aggregation, okay, so what by pushing these reactants like that, they collide with each other and makes the reactions to go faster like that. This was the explanation given by Breslow, okay, in the year 1980. And in addition to that, what happens is that salting out method, what has been used, lithium chloride, when it has been used, it further decreases the solubility of the reactants. Because why? Because it salts out, because salt is being added, and it decreases the solubility of the reactants further, because the water is being salted out, and further push, which further pushes the these reactant molecules to more. And because of this pushing effect created by the salting out of with the lithium fluoride, the molecules will collide even better and more, and thereby they get uh, reacted faster, and thereby we can get a better reactions. The products will be getting formed at the faster rates like this. Okay, so don't you think that it is all quite an interesting small discovery only, but still it has an it has such a significance take into the consideration. Till the Bristler discovered this reaction, we were using the organic solvents to drive these Diels-Alder reaction. After Breslow discovered this reaction using in water, now everyone follows this water as a solvent for this Diels-Alder reaction. Like that. Okay, so that is how the problem. Simple, small modifications here and there when we are doing any reactions in the terms of in chemistry, we can get a big results out of that. Okay, on water chemistry, if I want to continue on this, okay, on water, if you take into consideration, then on water reaction, okay, insoluble reactions are studied in water as a suspension like. So that means using in water, what happens is it will becomes organic compounds are relatively non-polar, and water is polar, and they what is the solubility slogan says, like dissolves like. Okay, So here it is like and unlike. So what happens is water doesn't tend to solubilize much. It will be like in suspensions, and these suspensions okay, uh, stirred as in suspensions to drive these chemical reactions. So these suspensions will give faster rate and better yields. Okay, so this was being given by the Sharpless in the year 2005. Okay, so this was a famous and this, in terms of epoxide, if we take into consideration, epoxide with piperazine on water take into the reaction. This is an epoxide. You can see that epoxide is there, and this is an piperazine derivative. And piperazine has got NH functionality here. If we react with an epoxide, what happens is it makes a covalent bond, and thereby this O gets converted to the O, and then this H migrates to the oxygen and it becomes an OH like this. This is the reaction. So now what will happens here is that under the need conditions, the time will be around 72 hours it will take, and the yield is 96 yield it will be under the need conditions. Need conditions means without using any solvents like okay. So this is also good, but when it is being used toluene, the time taken is 120 hours it takes, and the yield is less than 10% of the yield when it has been taken an organic solvent like toluene if you take into consideration. If you take ethanol, the polarity increases, and because of the compared to the toluene, and the time taken is just 60 hours when in comparison, and the yield is around 89% of the yield like that. Okay, but when the water is being used as a solvent, you can see that time is 12 hours, and it takes 88% of the yield is being obtained for this particular reactions. Okay, so that such is the importance of this green chemistry, or using in water as a solvent. Water has got a high potential to be used as a solvent in many of the organic reactions that we should understand, and we should try to do reactions in water. We should try to do, if it comes, it will be really good and handy. So now the next lesson is that is the water is polar in nature. Okay, as we say, suppose if it is an dipole moment of close to 1.8, it is so enormous. Okay, so you can say if you take the structure of an water molecule, okay, the water molecule structure is poised in such a way that just like oxygen and two hydrogens like this. So the the center of positivity and center of negativity are slightly distant, and intensity of the charge is also high. So what happens is dipole moment will be certainly high. So since the dipole moment is high, it is said to be an polar in nature from the textbook lesson if we take into consideration. Okay, so but what happens is that is water is polar in nature? If that is a definite question, then the answer is, the actual answer is, it depends. It not necessarily to be a polar in nature. It depends. It depends on what? It depends on the temperature. This is very important. So now, if you take water is polar at room temperature. At room temperatures, if you take into consideration at 20 degrees, 25 degrees, 27 degrees Celsius. The water is polar in nature. Okay, but what about its polarity at higher temperature? If we take in consideration, at higher temperature, okay, the polarity decreases. The hydrogen bond breaks. Okay, as the temperature increases, the temperature overcomes these hydrogen bonds, which is strong electrostatic force that holds the two molecules of water together in the crystalline structure of an ice if you take into consideration, or in the liquid state. So once these hydrogen bonds have been overcome because of the heat that we are supplied, so what happens is it will becomes once these hydrogen bonds are out of the touch, out of the intact, then the water becomes more polar in more non-polar in nature. From polarity it turns to the non-polarity. Okay, so how does it? That is quite an interesting one. If you take it at temperatures of 200 degrees Celsius, it resembles the organic solvents. At temperatures of 200 degrees Celsius, if you take water, okay, it resembles the organic solvents, how they are in terms of polarity. So like that, the water will be, okay. Now if you take at 300 degrees Celsius temperature, okay, it resembles the polarity of an acetone. We all know the what is the polarity of an acetone. Okay, so it resembles the polarity of an acetone at 300 degrees Celsius temperature. My dear colleagues, my dear friends, this is the reason why you study in the in-silico design. When we do molecular dynamic simulations after doing the docking, how stable the interactions are, how stable the interactions are at different conditions. In that conditions, the temperature is also one of the important conditions, because these interactions will differ based on the temperature taken into the consideration. Okay, another very good example what I can give it is, if you touch this water, if just your touch experience with the water in the winter season, how the water will be intact, how the water will be hard and intact. Okay, but if you touch the same water in the summer, hot of the summer, how the water you can feel it, and uh, you can feel it, it is completely different. So that is depending upon the temperature, how the water behaves, and with that what we can feel with the water on site will be different. Like, okay, so at high temperatures, water has lower density and is a stronger acid and a base. Okay, so at high temperature what happens is water has lower density, it gets reduced, and it's a stronger acid and the base, being amphoteric in nature, water. So this is the understanding that we will be getting it. Okay, so the next lesson is that dehydration reactions ought to be done in anhydrous conditions. Okay, whether the dehydrations, many of the reactions, what we feel is that whenever we are doing any of these reactions, these especially the if the reactions involves an dehydration reactions, then these dehydration reactions we have to carry out in the anhydrous conditions, whether we need to carry out in the anhydrous conditions. Okay, to unlearn that, not necessarily. Okay, so remember, even though it is a dehydration reaction, because water is a product here, so if we can eliminate that product, okay, so then this if there is a reversibility of the reaction, if it is the reaction is attaining an equilibrium or a reversible reaction, so then reversing back to the reactant can be reduced. So that was the concept. Okay, suppose if it is a non condensation on the active methylene group, if you are reacting aldehyde or ketone with an or okay, with an active methylene group, and then there is an water is getting eliminated, so we have to remove that water, and so that by using Diels-Alder operators, anything like that, so that we can shift the balance towards the product formation, so that the product combining with the water will not be there, so thereby bouncing back to the reactants will be prevented. Like that. So no, not necessarily like that. Even some of these dehydration reactions, okay, the truth is that, okay, it need not to be conducted in the absence of, not to be conducted in the absence of water, it can be conducted in the presence of an water. Like we can use water as a solvent for these dehydration reactions also. In what way? If you take dehydration reactions need not be conducted in the anhydrous conditions. Okay, it is doesn't require some anhydrous conditions. Okay, need not be conducted in the anhydrous conditions. It can be conducted in water. This is the understanding. This is the lesson that we can learn. Okay, this is the one false belief that we have, is that dehydration reactions needs to be conducted in the anhydrous conditions. So that we should overcome, and the understanding is that it can be conducted in the water as a solvent. We can use water as a solvent. Okay. So now with this, I'll conclude my presentation. So, my dear students, that my dear colleagues, my dear friends, is that you can see that this is the nature, how it looks like. Okay, in this slide what you are seeing it, see in this nature is the best model, in the sense that what happens in this nature with respect to the biological system, if we take into considerations, we tend to mimic how they are the in the nature, how these organic molecules or biomolecules exist. Okay, on the computer screen. If you are doing an in silico drug design, okay, molecular modeling, so what is your ultimate aim is to mimic how these molecules that tend to exist in this nature, like that. That's how you are trying to mimic it in the real-time environment on the screen, uh, like that. So now the question is that, are you good at mimicking how they exist in the nature onto the computer screen? Suppose you may be doing an QSR, you may be doing an, for example, you may be doing an docking like that. You need a ligands, you need a proteins like that. Are you sure that what are the molecules that you have drawn and you have converted that to a three-dimensional structure by the various models that you are using it? Are you sure that that is the exact three-dimensional structure? That means whether your modeling is right. Are you sure about that? Okay, the answer is many of the times we will not be sure about the three-dimensional structures of these ligands and the proteins what we use it in our in silico drug design. Because when you are trying to do the energy minimization, when you are trying to minimize the energy to convert two-dimensional structure into the three-dimensional structure, there are lot many tools are available, lot many methods are available. Okay, now the question is that which method, which tool to be used effectively to get the actual three-dimensional structure of the compound or the protein, like. So the ligands that we use it from our synthesized compounds, if we don't know the hexacristallographic structure of that single crystal structure of that three-dimensional structure of that compound, if you don't have, then if you assume that whatever by energy minimization, that is a three-dimensional structure, you have minimized the energy, no guarantee that that is the exact three-dimensional structure of that particular compound it is being. So then, in case if we are using that, we need to know that how it exists in the nature, like that. Okay, whatever the analysis you do, whatever the tools you use, whatever you perform, okay, if it is not correct, it is everything will be incorrect. It goes wrong, as simple as that. So that is why, even though we have lot many computational tools to use them effectively, okay, one should be trained, and one should use it properly with this knowledge in the background. Okay, so that we can become an effective modelers or effective in silico chemists, we can become like that. So this is not that easy if we take into the consideration. There's a big challenge here Okay, because to mimic the three-dimensional structure of the molecules, the way how they exist in nature, it is very very difficult, because it is one in a thousand possibilities if you're doing it by manual method, automated method, if you're doing it like so. The nature is the nature actually, that's very important. Okay, so we should, our attempt is to make the things how they exist in this nature, like that we are trying to mimic them, like, okay. So like this, and I acknowledge Dr. Emmanuel Rob from Diels Lab for the concept and some of the slides presented in this presentation. So I greatly acknowledge Dr. Emmanuel Rao. It was his concept and his okay. Uh, and this presentation, it was on this topic of learning chemistry by unlearning and then relearning it again freshly. Like, and also, I'd like to thank Dr. G. Jagdish of USFDA for motivation and the support that he renders to me on the time. And um, as I got, and this is our college, JSS College of Pharmacy at Mysuru in Karnataka. Ours is an NIRF ranked 10th in the country, and we have uh, most of the branches in pharmacy in as an inform program, and also the PhD programs here. So in case if you are interested, the campus here, the campus life, the environment is electrifying. Uh, if you come into this environment by any chance for any seminar, conference, or workshop, you can always welcome. You can come like that. So I wholeheartedly thank you all for your patience listening.